All right, I think we'll go ahead and get started. Good morning, everyone, and thank you so much for joining the webinar today. My name is Emily Knowlton, and I'm a Senior Immunology Sales Specialist with ProImmune. I am based in Pennsylvania in the United States, and I am an immunologist by training. My PhD work focused on evaluating both T cell and B cell responses to human herpes virus 8 infection, which is an oncogenic virus that causes Kaposi sarcoma. And today's webinar will focus on the ProPresent antigen presentation assay, commonly referred to as MAPS, which stands for MHC-associated peptide proteomics assay. And before I get into the details about the service, I do want to briefly introduce ProImmune, the company, for those of you who may be unfamiliar. We were founded in 2000, and our headquarters are in Oxford in the UK, though we do have a presence in both the US and in Singapore. We're an ISO accredited lab, meaning we have high quality standards for the reagents and the services that we offer. And our customers are essentially anywhere immunology research is being done. So that spans more than 50 different countries. We've worked with all of the top 10 pharma companies, including well-known biotech and academics as well. And some of those publicly disclosed clients are shared on this slide. And we were founded with a focus on providing MHC multimers to track antigen-specific T cells. And we've since expanded that expertise to provide many other immune monitoring tools, as well as epitope discovery and immunogenicity assessment services as well. We've also recently launched our Anchoron technology, which are small target binding proteins. And you can learn more about those on our website or in our Anchoron webinar. But again, today's focus will be on ProPresent, which is a highly sensitive and really exquisite tool in identifying naturally processed and presented peptides from a protein of interest using mass spec. So this broadly applicable service can be used to characterize immunogenic regions of a biotherapeutic, for example, that can lead to unwanted immune responses or immunogenicity, including addressing the drug's mode of action or evaluating the impact that protein modifications have on peptide presentation. Now, in the context of a wanted immune response, ProPresent can also be used to identify novel epitopes and new targets for vaccine and immunotherapies, which I'll talk a little bit about today as well. The turnaround time for ProPresent is very quick. In around 6 weeks, we can assess class 1 and or class 2 peptides, just depending on the project focus. And we have an unrivaled client publication record. And I'll be sharing several of these as case studies today to demonstrate the wide array of test articles we've assessed, including antibodies, blood factors, gene therapy vectors, oncolytic viruses, and even food proteins. So as I mentioned, MAPS assays are widely used in immunogenicity assessment to characterize immunogenic regions of a biologic. They're really considered the gold standard in the field for this purpose. The formation of anti-drug antibodies, or ADAs, can occur via a T-cell dependent mechanism, which is shown here in this cartoon, where the therapeutic gets taken up by an APC, processed, and then presented via an MHC class 2 molecule to a helper CD4 T-cell. Those activated CD4s can then interact with their cognate B cell, resulting in the proliferation of plasma cells and the release of ADAs. And those ADAs can alter the PK and the PD of the drug and ultimately result in treatment failure. So each assay in our immunogenicity risk assessment toolkit, which is shown here in this table, is designed to address key questions along this pathway and provide information to help make decisions about your program. So identifying what sequences from the therapeutic may be treated as foreign is a very important step in understanding immunogenicity risk, and this can be accomplished using ProPresent. As you can see in the table, it does not tell you anything about T cell function. So it is commonly used in combination with functional T cell assays, like our peptide-based ProMap T cell proliferation assay or our whole protein-based ProCern DCT proliferation assay. So for the ProPresent, we use our ProImmune PBMC biobank. So these are PBMCs that have been collected from healthy altruistic donors in the UK. They are fully typed for class 2 and then cryopreserved. So we'll select a broad panel of donors to fit the global population for each study. And if there's a specific allele of interest, then we can overrepresent that as well. Now, a typical project size for ProPresent is between 10 and 20 healthy donors. So we use those PBMCs to generate monocyte-derived dendritic cells, and then we load exogenous antigen onto those cells. So that can be something like a bispecific antibody or a therapeutic protein, for example. But those DCs then process the antigen and upregulate class II on the surface with those peptide epitopes bound. We then lyse the cells and then do an immunoprecipitation step, pulling down the MHC peptide complex. And here we can use either an anti-HLA-DR, DP, or DQ antibody, just again, depending on what the focus of the project is. We can recover the peptides and then sequence them using mass spec. So we'll create a database that has the human proteome and then the sequence from the input protein. And we probe that database with all the mass spec peptides that were identified. So the data might look something like this with your donor ID, their allele expression, any sequence that they presented, as well as an expect value, which is indicative of peptide identity and is a measure of predicted mass of the peptide versus the mass spec measured mass. So it's similar to a p-value in that the lower the expect value, the more confidence that you can have that that peptide masses are aligned. So the expect values of less than 0.05 are highlighted in blue here. So as an example, donor number 11 expresses DRB1, 0301, and 0401, and they presented two unique peptides, one from the variable light chain and one from the variable heavy chain. And you can see with the heavy chain, there are actually several fragments of the peptide within that nested region between amino acids 77 and 93. So when you look at the data collectively across all of the donors in the study, you can start pinpointing those epitope clusters and promiscuous binders. So this DNA KN sequence was actually presented by every donor in the study that presented a portion of this protein. And that ranged from 1 all the way up to 6 different fragments. And of course, these individuals presenting this peptide all express different alleles. And so it's really these promiscuous peptides that can be presented by a range of donors with different HLA types that could potentially be at higher risk for immunogenicity due to that broad population coverage. And these are certainly the peptides that you would want to further explore in functional T-cell assays like the ProMap. So the first case study comes from Editas and the FDA, where they used ProPresent to characterize CRISPR-Cas9 immunogenicity. And CRISPR-Cas9 is a gene editing tool that offers unprecedented opportunities for treating disease. However, Cas9 is of bacterial origin. So any unwanted immune responses against the bacterial enzyme would have, of course, serious implications. So to better understand the immunogenic regions of their Staphylococcus aureus Cas9, we performed an 18 donor ProPresent study, and the alleles of those individuals are shown on the y-axis with the position on Cas9 on the x-axis. Each different colored blob represents a different donor, and the size of those little blobs represent the number of fragments at that position. So you can see that there are several alignments across Staph-Cas9 where multiple donors presented regions of peptides from that area. And overall, there were 22 unique peptides that were identified in ProPresent that also induced T-cell proliferation as there were subsequent T-cell assays performed. And that data set's overlaid in pink on top of the ProPresent data. So that equates to about 85% of the peptides that were processed and presented that also induced T-cell responses. Now, to give you an idea of the scope of a HLA coverage for this size of project with 18 donors, this is about 70% of the North American population, with between 2 and 10 unique peptides being presented by each individual. So this certainly emphasizes the importance of including a wide range of donors, as well as any new Cas9 variants that are being explored. It's very important to consider these types of approaches to better understand that immunogenic potential. So one of the most pressing questions that were asked about MAPS is how predictive it is in reflecting clinical outcome. And while there are multiple complex factors that can contribute to clinical immunogenicity, this is an excellent example of assessing sequence-based risk. So this is a retrospective study with Novo Nordisk and the FDA on Vytreptocog alpha, which is an engineered factor 7A analog. And the native recombinant factor 7A has been used for more than 20 years with no reports of clinical immunogenicity. So they incorporated 3 amino acid substitutions to increase that enzymatic activity. maintaining more than 99% homology to that native sequence. But when they entered into the phase 3 trial, there were ADAs observed. And so Novo Nordisk had to discontinue the drug development at that very late stage, which of course is quite costly. So they wanted to use some of these preclinical assessment tools to see if they would have predicted that they had engineered in these neoepitopes. So we did a 12 donor per% study, and then they did a 17 donor MAPS assay in-house. These are different donors, completely independent studies that yielded essentially the same results. So we looked at presentation of recombinant factor 7A as well as Streptococcus alpha. And then the 3 amino acid substitutions in those protease domains are shown above each study outline, with a V to D, an E to V, and an M to Q mutation. And all of the peptides that were obtained were detected over these E to V and M to Q mutations. They did also go on to do T-cell proliferation assays with these mutant peptides and showed that they could activate or cause CD4 proliferation. So even though there were only 3 amino acid substitutions incorporated, 2 of them were processed, presented, and activated T cells. So immunogenicity was determined as the root cause of failure for this drug, which highlights the importance of stratifying your patient data by HLA type, especially with sequence-modified drugs, and also provides further evidence as to why these preclinical assessment tools are so important to use. So here's another example of sequence modification of therapeutics. This is for factor 9. And this is data from Catalyst Bioscience, in which ProPresent was used to characterize peptide presentation from DALK-A. And DALK-A is their next-gen factor 9 for treatment of hemophilia B that was developed using rational protein design with 3 amino acids changes noted here. And this results in a 22-fold enhanced potency of the drug compared to wild type, which is Pfizer's Benefix. And that's been on the market for more than 25 years. So during their phase 1/2 trial, 2 patients did develop ADAs that did not cross-react with the wild type factor 9. So they performed a very comprehensive comparison of those 2 proteins. So they began with in silico analysis on DALK-A, which did predict low immunogenicity risk. And then we performed a DCT cell assay showing comparable low responses and frequency of stimulation between DALK-A and the wild type. We then performed the 12 donor per% study looking at DR, DP, and DQ presented peptides. And you can see very similar peptide presentation profiles in the conserved regions, with actually a reduced number of donors presenting those mutated peptides from DALK-A, which are summarized in the pink table to the right. They did also show a low overall response to DALK-A derived peptides in a ProMap T-cell assay that were performed. So overall, they concluded from this comprehensive analysis that the DALK-A showed no increased risk compared to the wild type factor 9. This next case study is from CSL Behring and the FDA, who used ProPresent to characterize peptide presentation on factor 8, which is a replacement therapy for treatment of hemophilia A. And most of the marketed factor 8 therapeutics fall into 3 main categories, including human plasma-derived, full-length recombinant, and then beta-domain deleted recombinant factor 8. And the originally approved full-length recombinant version products do appear to be more immunogenic based on observational studies and prospective clinical trials. So they hypothesized that the platelet-derived has fewer T-cell epitopes. So we performed the ProPresent study for the full-length recombinant factor 8, platelet-derived, and then Afstilla, which is their beta-domain deleted version, all in the presence of von Willebrand factor. And you can see there are many more peptides presented via DR, DP, and DQ in the full-length version. So something very interesting is happening during this synthesis process that's essentially changing the peptide presentation profile of the recombinant factor 8. And you can hear Dr. Zubin Sana present this data from one of our Mastering Immunity conferences, and that's on our website. This study does also address a couple of other key questions that were often asked in regard to MAPS. So in figure B and C, we see the presentation of the full-length recombinant factor 8 and platelet-derived factor 8 in the presence of von Willebrand factor. And then the number of peptides at those positions are on the y-axis. So not only did they use the healthy donor cohort that we would typically use, and that's shown in blue, but we also looked at a subset of hemophilia patients, which are shown in pink. So the magnitude or the number of peptides presented do differ between the two cohorts. However, you can see that the peptide presentation profile is nearly the same. So this provides further support as to why healthy donors are typically used in these preclinical assessments. And then in part D, you have the number of peptides presented from the full-length protein. And of course, factor VIII is very large, and so you certainly expect a large number of peptides to be presented. And as expected, there are nearly 1,800 DR presented peptides, which is what most groups will focus on. But they did also look at DP and DQ, and you see a total of 500 other peptides that are being presented. So these could be potentially immunogenic and missed had they not looked at all three of those loci. So the case studies thus far have focused on presentation of peptides that can lead to ADA, but it is also true that presentation of T-cell epitopes is a prerequisite to generate regulatory T-cells that can contribute to oral tolerance. So this is a case study with Danone and how they used ProPresent in the identification of peptides with tolerogenic potential in a hydrolyzed whey-based infant formula. So one of Danone's products is infant milk formula, and one of the big challenges is that cow's milk protein is the leading cause of food allergy in children under 3. And that's likely driven by the fact that they didn't generate oral tolerance to those proteins. So the typical treatment for this is to use hydrolyzed cow's milk-based infant formula because it's been shown that allergenicity decreases with decreasing protein chain length. And that is shown here in this figure, where you have the regular formula, which includes the full intact protein, a partial hydrosylate, which is the prevention formula, an extensive hydrosylate, which is for mild to moderate allergy, and those with a severe allergy, the only option are the amino acid components. So in this case study, Danone was interested in identifying what peptides are being presented from this prevention formula that could potentially be tolerogenic. And the protein that Danone was most interested in was beta-lactoglobulin, or BLG, which is the most abundant component in whey and one of the major allergens in cow's milk formula. So we performed a 12-donor ProPresent study, and there were 2 main sequence groups that were identified from BLG, including fragments between positions 11 and 30, and then 23 and 39. So there's several donors presenting multiple fragments from each of these regions. And interestingly, both of these regions overlap with a portion of BLG that had previously been shown to reduce allergic skin response in animal models. So these certainly are potential candidates to induce tolerance. So we've seen a number of different examples of ProPresent being used at different stages of drug development. How and when you use MAPS is something we are often asked, and those answers are going to be project and company specific. Some groups will incorporate MAPS early on in the drug development stage to help design less immunogenic scaffolds, for example, whereas others might already know a protein is immunogenic and they need to better understand what they can do to reduce that immunogenicity. So one strategy that has been successful for our customers is illustrated here. Once a lead candidate has been selected, we can identify the immunogenic peptides in that lead using ProPresent. We can then synthesize the peptides for you and evaluate them in the ProMap T-cell proliferation assay, not only to confirm T-cell responses, but also to help rank those epitopes in terms of immunogenicity risk. So the combination of these assays as part of a phase 1 can provide potential reengineering options and opportunities to be explored. So there's a really nice talk on our website by Mike Zimmer from Moderna that talks through Moderna's experience in going through this phase with us and their future plans to de-risk their molecules. So companies can then move on to the de-risking portion or the reengineering phase, where they can try to disturb the anchor residues of those epitopes to prevent or reduce binding to MHC. And then those second-gen candidates can be expressed so that the protein functions can be assessed and binding to the targets can be confirmed. And then you can take those re-engineered versions into kind of a third phase and directly compare them to one another, as well as to the wild type sequence in our DCT assay for whole protein antigenicity. And if everything is successful, then you would have a candidate that does have reduced antigenicity compared to the wild type that maintained its therapeutic potential. So just to switch gears a bit on my last couple of slides, as I mentioned earlier, ProPresent can also be used to look at class 1 presented peptides, but because this is a different processing pathway and the dendritic cells we use in our standard method are not great cross presenters, we do modify our approach slightly. So rather than generating dendritic cells and then loading exogenous antigen, we would work with cells that have already seen the antigen in the class 1 pathway and are submitted to us by our clients. And those cell sources can be tumor tissue or tumor cell lines, healthy primary cells, treated patient samples, just essentially anything that has class 1, which is nearly everything. And then the antigen source can be DNA, RNA, viral vectors, tumor lysates, and you'll hear a couple of examples of those in just a moment. So as a first step, the customer prepares the antigen experience cells by pelleting down and freezing, and then the work at ProImmune begins with the lysis and immunoprecipitation step, which we would use a pan class 1 antibody for. So these next two examples demonstrate the importance of identifying those class 1 restricted epitopes through this method for use in the immune monitoring phase of their respective programs. So this is from the Children's Hospital of Philadelphia and how they use ProPresent to identify adeno-associated virus-derived peptides in their gene therapy program. And of course, AAV is commonly used as the vector for gene therapy. But given the high proportion of adults who have neutralizing antibodies against AAV1 and 2 serotypes, it's very important to understand and then monitor the cellular immune response against AAV, also to ensure a long-term successful gene transfer strategy. So the purpose of this study was to identify class 1 restricted AAV peptides that could be used during the immune monitoring phase. And they provided us with HHL5B7 hepatocytes that were transduced with AAV2 encoding the viral capsid alone or with the human factor IX. And then we identified peptides presented from the capsid protein. So this included this SLD sequence from the AAV2 capsid, which is highlighted in yellow. This was presented by both samples and had low expect values. So they had these peptides synthesized and then went on in an ELISpot assay to confirm that this SLD peptide does trigger a cytotoxic T cell response. And then lastly, this is a case study from the German Cancer Center and Orix, which is an oncolytic virus company. And this is the use of ProPresent in the identification of epitopes from their oncolytic parvovirus therapy. And oncolytic viruses are a very promising immunotherapy that helps to turn on the immune system by selectively replicating in cancer cells to induce specific anti-tumor immunity. And this study was done with a rodent protoparvovirus, H1PV, which is non-pathogenic for humans. It's very small and it's highly oncoselective. And they were exploring their PARV-ORX-01 therapy in a phase 1/2a trial to treat glioblastoma multiforme, which is the most common primary brain tumor with a very poor prognosis. And they needed to identify relevant parvoviral and tumor epitopes to monitor responses to the treatment, but didn't have enough cells to do both the epitope discovery and the immune monitoring because, of course, these patients are quite sick. So they provided us with a parvovirus-infected glioblastoma cell line that had been matched to the patient's HLA type, and then we performed the pan class 1 ProPresent, in which 5 peptides from 2 different parvoviral proteins and 20 peptides from 18 different glioma proteins were identified. So this allowed them to then design peptide pools containing both literature identified and ProPresent identified novel epitopes that they could monitor the T cell responses against in an ELISpot assay that we performed. So thank you so much for your attention. That was a lot of data, a lot of really interesting studies from our customers. Please do reach out to myself or our inquiries email address, which is listed below, to discuss your project and this assay in more detail. With that, I am happy to take any questions. You can either just raise your hand and ask your question verbally, or you can type something into the chat box. Thank you. All right, well, everyone's thinking of those really great questions. This is just a small summary of where we're going to be over the next couple of months. So if you plan on being in attendance at any of these conferences, we're either going to be doing posters or exhibiting or providing talks. So please stop by and say hi to us. Any questions? Nothing in the chat just yet. Also with this assay, as well as any of our other assays, you can request sample reports, which detail the methods of the assay as well as how the data is provided. Okay, well, it doesn't look like there are any questions. So thank you again so much for joining, and I hope everyone has a great rest of the day. The webinar will be posted on our website probably within the next week or so, so just hang tight. All right, thank you.